I don't always listen to podcasts, but when I do, I listen to STEM Talk, interviewing the most interesting people in the world of science and technology. Stay curious, my friends.
Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society. Hi, I'm your host, Dawn Carnegas, and today we have Dr. Dale Bredesen with us, a physician and scientist who is conducting extraordinarily innovative research on Alzheimer's disease. Also joining us today is Dr. Ken Ford, IHMC's director and chairman of the Double Secret Selection Committee that selects all the guests to appear on STEM Talk.
Hi, Dawn. Good to be here with you.
So Dr. Bredesen is an internationally recognized expert in the mechanisms of neurodegenerative diseases such as Alzheimer's disease. The continuing failure to find effective treatments for Alzheimer's disease has highlighted the critical need for a deeper understanding of the probable multiple causes of Alzheimer's disease. Dr. Bredesen's innovative work has led to new therapeutic approaches to treatment of this devastating disease.
Dale's group has found evidence that Alzheimer's disease stems from an imbalance in nerve cell signaling. In the normal brain, specific signals foster nerve connections and memory making, while balancing signals support memory breaking, allowing irrelevant information to be forgotten. But in Alzheimer's disease, the balance of these opposing signals is disturbed. Nerve connections are suppressed, and memories are lost.
And this model is actually contrary to popular belief that Alzheimer's is a disease caused primarily by the accumulation of plaques in the brain. In contrast, Dr. Bredesen characterizes Alzheimer's as a metabolically driven neurodegenerative process. But before we get to today's interview, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we especially appreciate all the wonderful 5-star reviews that are piling up on iTunes.
As we announced in several earlier episodes, the aforementioned Double Secret Selection Committee has been continually and carefully reviewing the iTunes reviews with an eye toward selecting the best reviews to read on STEM Talk. There are extra points given for pithiness, wit, and of course, lavish praise.
If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk shirt. Today, our winning 5-star review was posted by someone named Mark Riff. As soon as we hear from Mark, a STEM Talk shirt will be on the way to him.
Here's Mark's winning review. Fantastic lineup, and what a wealth of cutting-edge information. Just having access to these incredible minds is unbelievable. Can't wait to see what's coming up.
Again, thank you, Mark Riff, and the thousands of other STEM Talk listeners who have helped make STEM Talk a success right out of the gate. Okay, so now on to today's interview.
Fasten your seatbelts.
Dr. Bredesen graduated from Caltech, then earned his medical degree from Duke University Medical Center in Durham, North Carolina. He served as chief resident in neurology at the University of California, San Francisco, before joining Nobel laureate Stanley Prusner's laboratory at UCSF as an NIH postdoctoral fellow. He held faculty positions at UCSF, UCLA, and the University of California, San Diego. Dr. Bredesen directed the program of aging at the Burnham Institute before coming to the Buck Institute in 1998 as its founding president and CEO.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Dale, welcome to the podcast.
Good to be here. Thanks for having me.
Yeah, thank you so much. So, Dale, can you just give us a little bit of background about how you first got into the type of research that you're doing?
Yes. So I was an undergraduate at Caltech, and I was very interested in chemistry and in the brain functions related to behavior, learning, and memory. And I then went to medical school because I was interested in understanding how diseases actually affect the brain and alter things like learning and memory. And I wanted to kind of put the two together and understand what is the chemistry that drives the changes in our learning and memory that occur with things like Alzheimer's disease and frontotemporal dementia and Lewy body disease and things like that. So that's how I originally got interested in this. And of course, with the whole molecular neuroscience revolution of the 1980s and 90s, it really offered us tools. And of course, that's continuing to offer us novel tools to understand these diseases in ways that we've never understood them before. And as you know, the whole area of treating and reversing neurodegenerative conditions like Alzheimer's disease and Huntington's disease and Lou Gehrig's disease, this has been arguably the area of greatest biomedical research failure in terms of developing effective therapeutics. So I think that this is a very exciting time where for the first time we're beginning to see some truly effective therapeutics for these illnesses.
Yeah, it's a very exciting time, absolutely. And like you said, there's been a tremendous amount of time and energy and money that's been put into research focused on neurodegenerative diseases. And, still there's so much that we don't know, but it sounds like we're starting to develop a better understanding, at least at this point.
Absolutely. And I think that there are several things happening at the same time that are very exciting. I think that people are beginning to look at larger data sets and at more network systems biology types of approaches, which I think are going to be the way that we are able to have a major impact on these illnesses. Previously, of course, people were looking at single mechanisms. So, interestingly, people would spend their entire careers on one mechanism, like reactive oxygen species for Alzheimer's disease, or detergent effects, or metal binding, or, you name it. There are 20 more. And yet somehow, how is it that this disease is about all these things? Clearly, they all had data on which they based their assumptions and their hypotheses. And yet, nothing led to something that really had a big impact on the illness itself.
Yeah, absolutely. Well, so I guess we're just going to jump right into the research that you've been doing then. So in your recent publications, you've likened Alzheimer's disease to other disease processes, including cardiovascular disease and osteoporosis and cancer. Can you expand on what characteristics you think that Alzheimer's disease shares with these other disease processes?
Yes. So the idea of the analogy is that Alzheimer's disease is really a network imbalance that is very analogous, for example, to osteoporosis. So in osteoporosis, as you know, you have a set of signals that contribute to osteoblastic signaling and osteoblastic activity, where you have the laying down of bone. And on the other hand, you have a set of signals that contributes to osteoclastic, to taking up the bone. And for most of your life, of course, these are beautifully balanced, allowing you to have strong and well-formed bones. And on the other hand, as we age, it's often the case that this signaling is imbalanced. So in fact, your synaptoclastic signaling outstrips your synaptoblastic signaling for years, and you end up with osteoporosis. And what we found is that there is an analogy with Alzheimer's disease. You have ongoing synaptoblastic signaling instead of osteoblastic, and you have ongoing synaptoclastic signaling instead of osteoclastic. And there are dozens and dozens of inputs, and it may turn out to be hundreds. We don't know exactly how many it will be, but what the surprise was that when we looked at signaling from the Alzheimer's amyloid precursor protein, so-called APP, that you could see a beautiful balance here where you can have cleavage at three sites, the beta site, the gamma site, and the caspase site, that lead to 4 peptides that, interestingly, all support neurite retraction. They're essentially synaptoclastic signals. And on the other hand, you can have an alternative cleavage at the alpha site producing 2 peptides, SAPP alpha and alpha CTF, that both turn out to be molecules and peptides that give rise to synaptoblastic signals of producing and maintaining. And interestingly, they inhibit the cleavage at the beta and the gamma site on the other side. So APP turns out to function as a molecular switch where you can drive it one way or you can drive it the other way. And then we looked at, well, okay, what are all the things that actually feed into this signaling? And the surprise was that it's many, many things. And it's all the things that we know from epidemiology that are indeed critical for Alzheimer's disease risk. So interestingly, ApoE4, for example, is one of the things that alters this balance toward the synaptoclastic side. Estradiol, free T3, free T4, testosterone, exercise, sleep. I mean, on and on and on. Melatonin. I mean, you just go on and on and on. Dozens and dozens and dozens of incoming signals that impact this critical balance. And so that's why we argue that this is analogous to other chronic illnesses. And of course, we find ourselves doing a lot of arguing these days because we believe that much of the research on Alzheimer's disease has focused on a single mechanisms rather than on the network as a whole. And one of the things that's come out of our work recently is that Alzheimer's disease, for most people, is actually not a disease. It is a programmatic downsizing of the synaptic network, much like apoptosis or synaptosis. It's been described. And imagine that you have a company for a minute that has 10,000 employees, and you find that you are heading for the red. APP is essentially like your CFO who's looking at all the inputs, all these different molecules. It's literally an integrating receptor. And if you determined that you could not support a hiring of new employees, the first thing you would do is shut down that hiring so that you could keep the ones that you have. And so the first thing that happens in this programmatic downsizing that's referred to as Alzheimer's disease is that you essentially, go back to the company analogy, you essentially are taking away the temporary cubicles. You're not going to bring in new people, and again, literally, you're not going to store new memories. And if you think about that, if someone gave you a choice and said, well, you can either remember the Friends rerun from last night and forget how to speak, or you can remember how to speak and forget the Friends rerun from last night. Of course, you would want to forget the Friends rerun from last night. So the idea here is you've spent your whole life selecting the most critical knowledge and most critical learning. You're actively forgetting things all the time. The 7 song that played on the radio, a week on the way to work a week ago, that sort of thing. And you're actively remembering the most critical things. So what we've discovered recently is that Alzheimer's disease, what's called Alzheimer's disease, is actually 3 different subtypes. And we just published this actually about a week ago. And these, if you look at the metabolic profiling, you can easily distinguish these 3. And 2 of them are literally programmatic downsizings, whereas the third of them is truly an illness.
Can you tell me where was that recently published, just so that we can reference that?
Yeah, it was published in the journal Aging.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
So yes, it's a completely different take on Alzheimer's disease. I mean, you hear so much about accumulation of plaques and tangles, and problems with clearance of some of those, some of that junk essentially that's in the brain. And essentially, you're saying that all of that stuff is kind of a side effect of something else that's going on.
Well, it's not a side effect as necessary, but it's a part of the mediator. So what happens is when you cannot support the extensive synaptic network, and you have close to 1 quadrillion connections, as you know, synapses in the brain. When you cannot support the addition of new ones, then you send out what is essentially involves memos. You send out 4 SAPP beta, A beta, which has obviously been the focus of a great deal of study, JCASP, which is the third peptide, and C31, which is the C-terminal peptide from APP. And these are all mediators of neurite retraction and literally of downsizing. So that, yes, when you see the collection of A beta and you see the collection of phospho-tau, what you're seeing is the downsizing memos. You're seeing the signaling that is, that is indicating that it's time to downsize. Now, that's important because if you simply get rid of the memo, you simply get rid of the signaling, yes, you'll buy yourself some time, but ultimately, you won't, unless you change the balance, ultimately you will have the downsizing. And so, no big surprise, the reduction of A beta has been very unsuccessful. And what minimal success there has been, has certainly not been any sort of major change in Alzheimer's disease outcome. It's been a very modest change, and in many studies, zero change.
So, my background is actually when I was doing my postdoctoral fellowship at Duke University, I was looking at apolipoprotein E and acute brain injury. And ApoE has been implicated very strongly in development of Alzheimer's disease. So how does that fall into the big picture?
Right. So ApoE has turned out to be extremely important, as you know, even going back to to the whole idea of evolution of the hominids from the simians. You had ApoE as being one of a relatively small number of genes that were altered in going from the simians to the hominids 5 to 7 million years ago. And of course, there is an overrepresentation of pro-inflammatory genes in that change. And Tuck Finch from USC has pointed out, I think in a very interesting way, that if you think about what it takes to change niches, to change from being arboreal to roaming the savannah and stepping on dung and getting wounds and eating more meat and fighting with each other and fighting with animals, you're really talking about the requirement for inflammation and for not only healing of wounds with inflammation, but the ability to eat microbes in raw meat. So in fact, it makes some sense that we would have pro-inflammatory genes as part of what allowed us to change niches and become what would ultimately be human beings. And of course, as you know, in antagonistic pleiotropy, the whole idea is that you have something that gives you an advantage when you're young, but ultimately gives you a disadvantage when you're older and limits your lifespan. And of course, with ApoE4, which is the only ApoE that's been around for hominids and for humans until only 220,000 years ago when ApoE3 first appeared, as you know, and then of course 80,000 years ago or so when ApoE2 appeared. So this has allowed us in evolution to be what we are, but has also, as you know, given us this markedly increased risk for Alzheimer's disease, for cardiovascular disease, and of course, it's also an anti-longevity gene. Of course, ApoE4 is underrepresented in centenarians, as you know. So what we found is if you go back, and one of the first things we did when we discovered this imbalance between the signaling on the 2 synaptoblastic side and versus the 4 peptides, the synaptoclastic side, is we said, well, look, if we're on the right track, then ApoE4 should have an effect on that balance. And we've published that in fact it has a major impact on that and increases the side of the 4 synaptoclastic peptides at the expense of the 2 synaptoblastic peptides. And more recently, we've been interested in how this works. And we found that, in fact, this has a major impact on the ratio of SIRT1 to NF-kappa B. So as you know, there's this very interesting mutual antagonism in a dynamic fashion between SIRT1 and its signaling versus NF-kappa B and its signaling. So literally, your cell is making a decision whether to put its resources into protection and essentially the NF-kappa B, activation of inflammatory processes, antimicrobial processes, etc., versus activation of more of a longevity, metabolic, more efficient way. It's almost like thinking about a country. Is it going to put its resources into war? Is it going to be more of a North Korea, or is it going to put more into development and research, more like South Korea? And what happens with ApoE4 is it alters that balance. It enhances the activity and the activation of the NF-kappa B, and it reduces the activation of the SIRT1. In fact, we see that when we looked at and published in PNAS, we looked in the brains, and this is the work of Ramohan Rao in the lab and Veena Thindakara. And so they've been looking at what happens with this ratio, and what they're finding now is that there's a very fundamental altering of cellular programming by ApoE4, by a mechanism that had not been appreciated before.
So that cellular mechanism was beneficial to our ancestors, but not necessarily to us now.
Exactly. And it's certainly beneficial to us now if we're living in a third world country, and if resources are limited, and if we're living in a squalid environment. It's going to be beneficial to us, and certainly very helpful during the first 40 years of our lives.
Mm-hmm.
However, late in life, and especially if we've been in an area where there are plenty of resources and there's not a lot of inflammation and the hygiene is good, then certainly in the long run, it's going to be a negative. And of course, interestingly, it also allows you to do well when there is limited food. And so, if you're going to die of starvation, you're actually going to survive better as an ApoE4 individual than as a non-E4.
Mm-hmm.
But on the other hand, if you're eating too much, which many of us are in the Western society, that's actually going to put you at risk, as you know, for things like lipid disorders and cardiovascular disease and cognitive decline.
Fascinating. So essentially, you guys are looking at these different signals that affect the ultimate molecular switch, which is APP. So you're looking at, am I correct?
Well, APP is a part of this, and actually we also just published information looking at the connectome that's involved with this plasticity balance. And it's interesting in that there are many, as you might imagine, many different connections. And yes, APP interacts with all sorts of different things that are related to synaptoblastic activity and synaptoclastic activity, including, by the way, of course, downstream effects on phosphotau and tau-related signaling.
Mm-hmm.
But clearly there are other things playing into this. For example, you need to have the appropriate building blocks. So things like specific lipids that are critical, and CDP choline, for example, and a number of other features. So yes, there are other things, but APP does play an important role in this phenomenon. And interestingly, because it is functioning as a molecular switch, this really tells us something about how the signaling works. If you think about it, we're all taught in school about homeostatic signaling, in which you have a negative feedback. And a simple example would be your serum pH, which you want to keep around 7.4. You never want it to be 10.4 or 2.4. So, if you drink an acidic soda and become a little acidotic, you have both metabolic and respiratory feedback that stabilizes you. On the other hand, there are many systems that require amplification and multi-goal outcomes, in which you require an amplifying step or steps. And a good example of these is in, for example, blood clotting, where, if you are a cave person and you cut off your finger accidentally, if you don't clot pretty quickly, you're going to die. So you amplify that signal rapidly, and you have a system of serine proteases that is actually a feed-forward system. It's a positive feedback. And so you see the same thing with APP. If you cleave to make the two peptides that are synaptoblastic, interestingly, they've both been shown to be inhibitors. In one case of beta secretase, in the other case of gamma secretase. So you're now actively inhibiting the APP from going down the synaptoclastic side. So what you can see, on the other hand, you go the other side and you're actively amplifying, and that's why A beta is really part of a prionic loop. So our argument is, in fact, that tells you something about the origin of prions in biological signal amplification. So when you have, it's a little bit like crowdsourcing. You go out, you are bringing things together to amplify a signal, just as you would, in fact, crowd synapses to amplify a signal. In this case, you're crowding A beta. And so this idea that these are aggregating proteins, and that this is some sort of mystery and involved with neurodegeneration because it's about misfolding. We would argue that, in fact, this is about biological signal amplification. And certainly in APP, you can see that very directly. And so no big surprise, yes, you want to lower the A beta because you want to lower that signal. But even more importantly, you want to lower the reason that that signal is occurring to begin with, which is because of the various inputs that are leading to this decision to downsize your synaptic network.
I think that's really interesting, actually. I was just reading a paper about, they were talking about protein misfolding and how it's actually a naturally occurring thing and it's transient, relatively speaking. And like you said, it's part of the signaling process. So I think that's interesting that you guys have kind of come up with this separately from your perspective.
Yes, and of course, I'm not arguing for a minute that proteins don't misfold. Of course, that's well documented. They do, and we've studied and published that for years. But my point is that there's more to it than that, that there is an active signaling. And by the way, this is no different than what happens in cancer. With cancer, you have the same sort of balance with tumor suppressor genes and oncogenes, and these are part of a very normal process. And on the other hand, if you have a mutation, if you alter the balance, reducing the tumor suppressor activity and or increasing the oncogenic activity, you end up with a runaway feed-forward process because you've got cells that actually have a Darwinian advantage and therefore survive more, divide more, etc., and you ultimately end up with a tumor. So you can actually select a relatively rare mutational somatic event by developing a tumor, which of course is the number 2 cause of death in the Western world right now. And as you've probably heard recently, the claim has been that the number 3 cause is actually Alzheimer's disease because it's probably been underestimated according to this recent work. So the idea is very much the same, that when you alter this balance because of this prionic loop feedback, you end up with a runaway process of essentially synaptic pullback and ultimately a programmed cell death. And if you look at the 4 peptides, that's what they mediate. They mediate loss of synaptic activity. They mediate pullback of neurites, neurite retraction. They mediate caspase activation. They ultimately mediate programmed cell death, and many, many other processes that are all related to that side of the plasticity balance, i.e. to synaptoclasis.
So this is a completely different approach to looking at Alzheimer's disease development, or as you call it, programmatic downsizing. So what led you to come up with this idea, with this approach to looking at Alzheimer's disease?
Right. First of all, years ago we were interested in what molecules mediate programmed cell death in the nervous system. Mm-hmm. And we discovered and published way back in 1993 that there is a new kind of receptor, and there are now over 20 of these receptors known, and there have been multiple international meetings on these, etc. And these are receptors that respond to the withdrawal of trophic support, whether you're talking about the withdrawal of nerve growth factor or BDNF or extracellular matrix support or hormonal support. You can go on and on. Any sort of supportive input and supportive signaling for neurons, and interestingly other cells as well. When you withdraw these, there is a set of receptors that we dubbed dependence receptors that mediate states of dependence. And so these things actually interact with caspases and signal, when you withdraw the trophic support, the activation of numerous molecules, but including caspases, and they signal things like neurite retraction and ultimately programmed cell death. And these things respond not only to trophic withdrawal, and one of the things that I was interested in then was that if you remember back in the 1930s, when Dirac had looked at the activity of the electron, he argued that perhaps there is an anti-electron, as he called it, a hole, which ultimately, of course, turned out to be the positron. This was the beginning. His prediction was of something that turned out to be antimatter. And so the question we had was, is it possible that there's also a trophic hole? Is it possible that there is an antitrophin? And at the time, there was nothing that fulfilled the criteria as an antitrophin. But it turns out that, in fact, amyloid beta peptide, A beta, fulfills all the criteria you would want as an antitrophin. So it interacts with numerous trophic receptors and it does just the same and induces the signaling that you would see from withdrawing the trophic support. So what we found is that there are numerous receptors that signal the presence or loss of trophic support. And these are related to, for example, there's been a lot published on netrin-1 and its receptors such as DCC, UNC5A, UNC5B, etc. And we found actually that netrin-1 also binds to APP itself. And so there is a dynamic balance between the netrin-1 trophic anti-Alzheimer's side and the A beta interacting. So interestingly, A beta interacts with APP and drives it down the side to make more A beta and more of these other synaptoclastic molecules. So that shows you immediately why it is that A beta is part of this preonic loop between A beta and APP. So this, again, is involved with amplification of signals. So when we discovered these dependence receptors, we started looking at what actually happens and do these things play a role in Alzheimer's disease. We found that A beta is part of this as an antitrophin, and by the way, it has antitrophic activities at the insulin receptor, as Dennis Selko showed. It has antitrophic effects at the alpha-7 nicotinic acetylcholine receptor and at APP itself, among others. So APP, as I said earlier, is an integrating dependence receptor over many of these trophic influences. So then we created transgenic mice. This is an early, we published this actually in 2006, showing that if you alter this balance, then you can have a major impact on the Alzheimer's phenotype in these mice. And if you just make a point mutation, for example, at the caspase site in APP, which doesn't alter the SAPP beta, for example, but it does alter the C-terminal ones, JCASP and C31, then in fact, you have a big impact on the ability of these mice to learn and on the ability of these mice to resist the Alzheimer's phenotype. And so this all supported the idea that there is a trophic-antitrophic balance that is out of balance in people with Alzheimer's. And of course, I wouldn't be telling you all this if we hadn't taken this into human beings and shown that, in fact, there are striking results in human patients. And we published the first 10 last September, where we can alter this balance in people and make them much, much better. And I'll give you a couple of examples. We have a woman who is the first one that came in actually 3.5 years ago now with clear signs her mother had died of Alzheimer's disease, and she had watched her mother go downhill. Her mother started when she was 62. This woman began to have symptoms when she was 65, very similar. She would get lost on the freeways, pretty classical symptoms. And she had trouble remembering numbers. She had to write down even 4-digit numbers. She could not remember what she read. She couldn't do her job anymore. She went to her physician, who told her that she had the same thing her mother had, and there was nothing he could do about it. He wrote down memory problems in her chart, and therefore she was unable to get long-term care, which is a common problem with people these days. And so she called her friend and said, "I'm going to commit suicide because I watched what happened to my mother, and there's no way I'm going to let that happen to her." So I got a call saying, would I talk to this woman? I said, "Look, I don't see patients. I'm a researcher, but I'd be happy to tell her what our research shows." And I'm happy to write a list for her, and she can take it back to her physician and talk to him. And of course, I thought I would never hear from her again. 3 months later, I got a call on Saturday at my home, and she said, "I can't believe it. I'm back at work full time. My memory's better than it's been in 30 years." And we also had, by the way, she ultimately had imaging showing that she indeed did have amyloid, and she's doing very, very well. Now 3.5 years out, basically back to normal. And we've seen this now repeatedly. We've also now have recent MRI quantitative data showing marked increases in volumetrics in regions of the brain with people who are on this approach. And the idea here is you don't just, if you've got an imbalance that has many inputs, you don't just try to tweak one input, you try to tweak as many as you can. And so we argued to patients that imagine you have a roof with 36 holes in it, because initially we knew about 36 different inputs to this network. And so you can fix one of those with a drug, which is fine, and that'll help a little, but it doesn't really address the underlying problem. And even if you go back to something as simple as HIV, which is much simpler than Alzheimer's disease, with HIV, as you know, it really took 3 drugs to have a significant impact. One by itself had a fairly minimal impact, but addressing all 3 had a big impact and ended up being the current way that works very well. It changed the landscape of HIV. So the idea is to address many of these. Now, as you can see from what we talked about earlier, this is a threshold phenomenon. So in this positive feedback model, in this preonic loop model, once you get far enough, it will flip over into the other state, because it's like a seesaw. You get enough on one side, and now you flip over. So this woman who started 3.5 years ago was only able to address 12 of the 36 things. But for her, at least at that time, it changed her so that she can now make new memories, store new memories. She now is doing very, very well. And she knows that ultimately it's possible that she will need to address others as she gets older, and we'll see. But for this first 3.5 years, she's doing very, very well.
Yes, I was going to ask, do you think this therapeutic approach will work for everyone regardless of what stage they're at with respect to Alzheimer's disease? Or there's some people that have gone almost too far and it's probably not going to have an impact?
Yeah. We don't know yet how long this will be helpful. We know that people who are in the SCI stage, subjective cognitive impairment, it's working very well. For people who are in the MCI stage, it's working well. And for people who are in the early Alzheimer's stage, it has worked for at least some people. In the late stages, we think it's unlikely. And the one person that on our first publication where it didn't work was in very late stages. And it really didn't have any impact, as far as we could tell, on her. So my assumption is that in the late stages, you've lost enough neurons that you're going to have to do something beyond this. But obviously, one of the research questions is, when you go as far as you can with this approach that we call MEND, M-E-N-D, which is for metabolic enhancement for neurodegeneration, what's the next thing you have to do? So when you get to the end of where that's going to help you, what is that next thing that's going to take this to the next level? So I think that's going to be a question going forward. Do you need stem cells? Do you need additional neurons? Do you need additional trophic factors that aren't provided? What is it that is going to be required for improvement at that point?
And then perhaps, just through MEND, creating an environment that is suitable for that growth that's necessary.
Exactly, and as you know, the whole story of stem cells and their role with aging and the milieu they're going to be in. So, what is it that you actually need? Certainly, you have some reparative capabilities in the brain. We see this all the time. The question is, how much can you put that to use when you are now trying to reverse Alzheimer's disease pathophysiology?
I don't always listen to podcasts, but when I do, I listen to STEM Talk, interviewing the most interesting people in the world of science and technology. Stay curious, my friends.
Can you give us some examples of the therapeutic interventions that you prescribe and that are part of the MEND program?
Right. So, first of all, people often ask, well, so just tell me the things to do. Well, it's not quite that simple. So, the point is that you have to start by asking, what is it that's driving the process to begin with? And as you know, the current hot area in oncology is genomic sequencing. And so the idea is you sequence the entire genome of the tumor and you sequence the entire genome of the person who is the host of that tumor. And that allows you to infer what the somatic changes are that are driving that tumor. And then you can choose appropriate therapeutics for that tumor. And of course, in the field of cancer therapeutics, they've been on to the idea of polytherapy since the 1960s. So what we're doing is no different, except that we have no tumor to biopsy. So on the positive side, we understand that Alzheimer's pathophysiology is really related to metabolic changes. So this is a metabolically driven neurodegenerative process. So what that means is we start by evaluating the person in as much detail as possible. What you really want to know is the genome and the metabolome. And with what's available currently, we start by looking at many different essentially metabolic profiling for these people. And of course, as you can imagine, this is not standard operating procedure, and so most physicians don't evaluate, even at Alzheimer's centers, aren't looking at most of these players, most of these mediators in the disease. For example, we want to know your copper to zinc ratio. We want to know your red blood cell magnesium. We want to know your inorganic mercury status and your organic mercury status in a very sensitive way. We want to know your free testosterone and bound testosterone. We want to know your estradiol to progesterone ratio. We want to know your pregnenolone. We want to know your homocysteine. We want to know your iodine status. On and on and on, because if you look at the inputs to that balance, those are the things, and dozens of others, that drive this imbalance. So if you, and by the way, I should say, for this profiling, we haven't found a single person yet who has fewer than 10 abnormalities when we do this metabolic profiling. And on the other hand, when we look at people who are asymptomatic, they typically have, few, 3, 4, 5. So all of the symptomatic people have between 10 and 25 abnormalities on this. The second piece is we don't want to drive you to the low end of normal. If you look at, for example, at a normal vitamin B12 level, it's between 200 and 900, but it's well documented that someone walking around with a B12 of 300 can certainly have problems. And so, ironically, one can die of B12 deficiency with a normal B12. That obviously is something that that should be considered semantically. So we want you to be optimal, not simply within normal limits. And so doctors use this shorthand of within normal limits, which when you're dealing with a degenerative process and a metabolically driven process can be a problem. And so we want to know where all these things stand. And then for each person, of course, you have a personalized response to this based on where you are with these different things. Fortunately, many of them can be addressed by metabolic alterations that essentially make you healthier. So, in fact, when you're looking at things like treating cancer, you're going to end up potentially with drugs that give you severe side effects. Whereas when you're dealing with these metabolic abnormalities leading to neurodegeneration, the first thing that happens is you're going to get healthier. And the second thing that happens is, and very interestingly, the cognitive function follows the metabolic function. And as you know, as an ApoE expert, you know that ApoE is a critical part of metabolism and plays a role, as we were talking about earlier, with sirtuin function versus NF-kappa B function, obviously with lipid carrying, binding to specific receptors and all sorts of downstream signaling for that. So the argument here, and Alzheimer's has been called type 3 diabetes by Dr. De La Monte, which I think is a very interesting observation. I think it goes beyond that. I think what we're seeing goes beyond just diabetes, but I think that the overall idea is a very good one, that metabolic status plays a critical role. And there's no question, and Dr. Getzel from UCSF has published beautifully that if you look at neural exosomes, in his study, 100% of people with Alzheimer's disease had signs in their neural exosomes of central insulin resistance, just as you would see with a type 2 diabetic peripherally. So I think there's little question. And again, as I mentioned earlier, Dennis Selkoe showed years ago that A beta interacts with the insulin receptor and actually inhibits downstream signaling from the insulin receptor. Mm-hmm. So again, we come back to this balance between the trophic and antitrophic activity, which is power, which is mediated by numerous inputs that relate to metabolism, hormonal support, trophic support, etc.
Mm-hmm. It's interesting too, because I've seen the studies showing impact on brain metabolism, even in MCI and obviously in Alzheimer's disease. So it makes absolute sense. That's really interesting. So essentially, you take all of these different factors and measurements, and then you put them into, is it a computer-based model that kind of develops the optimal program?
Yeah, we have both. So we have software that has been developed by a group out of Research Triangle. But we started obviously with just going by hand and just the simple old-fashioned way. Let's look at all these different things. But yes, I think that is where things are headed, mm-hmm. to larger and larger data sets. And if you take a step back and look at it, as physicians, we've spent thousands of years dealing with ridiculously small data sets. Here we're dealing with an incredibly complicated organism with 3.3 billion base pairs, and that's the simplest thing because that's just unidimensional. We're talking about a very complicated organism, and we know things like your sodium and your potassium. I mean, there's just so much that we don't know, and so what's happened? Physicians have had to be quote intuitive, good guessers, looking at the big picture. But for 21st century medicine, of course, with whole genome sequencing, with more and more proteomic availability, metabolomic, lipidomic, and more computational analysis, the future, of course, is for larger and larger data sets and less guesswork, mm-hmm. where we can now look and say, okay, look, here's what's causing the disease. And I have to say, it's been really striking to look at these people with early Alzheimer's and with SCI and MCI and look at their metabolic profiles. The first thing you see, as I mentioned earlier, is that it really falls into three different pockets. These are three different illnesses that we all refer to as Alzheimer's disease. And as I mentioned, two of them really aren't diseases. They're programmatic downsizing of your synaptic network. And so when you do this, you can see what's driving the process, and then you can alter what's driving it. You're really asking why instead of asking what. Is it Alzheimer's? Well, Alzheimer's is mysterious and untreatable. Sorry, go home and get your affairs in order. Mm-hmm. That's the 20th century approach. The 21st century approach is to ask why. Why did you get this? What's driving this? What are all your risks and what are all the metabolic drivers? And as you know, there's been a question for years. Why is it that so many disparate risk factors exist for Alzheimer's. You're at increased risk if you sit on the couch too much, or played in the National Football League, or have a low vitamin D, or went through menopause early, had an early oophorectomy, had a low vitamin D level, low B12. I mean, just go on and on and on. How are these things all related to each other? This is not a simple situation.
It's like cancer. Yeah.
Exactly. So they're all playing on this same network.
Mm-hmm. Very interesting. So, are there certain things that you think, it's like you said, you're collecting the data as you go through all this. Are there certain factors or certain inputs to the network that you think have more of an impact on the network than others?
Yeah, people always ask, what's the one thing I should do? And the answer is everything.
Yeah.
But the thing that's most important, I think, is to have these things evaluated for yourself so you know where you stand. If there are, I would say there's a few things that come up again and again as being important. And we're trying to look at, can we simplify this overall protocol? Because as you might imagine, one of the hardest things for the people who go on this approach has been to do all the different things. And they say, well, gee, this is not easy to do these different things. And so we have coaches and things like that help to walk people through it. And one guy said, could you send someone to my house who's a sergeant who just makes me go through my paces each day? So there are different ways, and by the way, this is nothing new for a cardiologist. If you went into your cardiologist and your cardiologist said to you, "Well, look, you've got atherosclerosis. I want you to stop the fries, but go ahead and keep eating the cheeseburgers and the muffins," you'd say, "What a lousy cardiologist."
Mm-hmm.
So we're doing the same thing essentially. We're saying here are the things that are driving you toward cognitive decline. And we can do many, many things to alter that balance and to make it so that we can follow you and drive you toward cognitive improvement. And that's exactly what we see with these people.
Mm-hmm.
But, you're going to need to do more than one of them. But to simplify, the things that come up recurrently are the following. Number one, as I mentioned earlier, Dr. Getzel showed that everybody tends to be insulin resistant in his studies, and we see that incredibly commonly in our studies. So that is an important one to address, and you can address it in numerous ways, pharmacologically and non-pharmacologically. So we need to drive people toward insulin sensitivity, and then after that to make sure that they have adequate insulin and related factors. The second thing that comes up again and again is various inflammatory mediators. And as I mentioned, NF-kappa B is a major player in this balance. And the third thing that comes up again and again is hormonal balance. And many hormones from estradiol to progesterone ratios to pregnenolone to testosterone to free T3 to reverse T3 to on and on. These are all critical players in synaptic formation and maintenance. The fourth thing that comes up repeatedly is exposure to specific toxins such as mercury. And it's interesting. Any one of these things, people will argue, this is not a cure for Alzheimer's. Well, no, but you shouldn't leave it out either. Any one of these things, again, you cover any one hole in the 36 holes in your roof, it doesn't do all that much. And so what happens is when you look at the entire group, any one of these things doesn't jump out as being the only thing. And so people say, well, we don't think mercury is that important. Well, for many people, it's not important at all. But there's a group out of New York called Sharp Again Naturally that has done some really interesting work showing that there are people, in fact, who respond to reduction in mercury with improvements in their cognition, people who have documented Alzheimer's disease. And of course, this has been controversial for years. And this kind of tells you why it's been controversial, because yes, in some people it's important, and yes, in many people it's not.
Yeah. So something like ApoE, like we were talking about before, we know that if you have 2 copies of the E4 allele, you have up to a 20-fold increased risk of developing Alzheimer's disease late in life. So, there's something like that to take into consideration. Are you looking at genetics as well with these measurements?
Absolutely.
Okay.
Absolutely. And we look at many others, of course, as well. And of course, ApoE is the most important of all. And one of the intriguing things, as you pointed out, there's a dramatic increase with ApoE homozygosity and a marked increase even with heterozygosity. And so, as you might imagine, we see just like everybody else, you see an increase in risk for those people. But if you look at these 3 different subgroups, 2 of them are largely programmatic. They're largely amnestic in presentation, and they select for ApoE4 positive individuals. The third group is really a fundamentally different disease, and it actually looks as if ApoE4 protects against that one. So there is an underrepresentation. At the very least, ApoE4 is not a risk factor, but it actually looks more like a de-risking factor. It is a different illness. Yes, it is amyloid positive, so it is grouped under the Alzheimer's group, but it is a fundamentally different illness. It presents in a different way. It presents typically in E3, E3, or even E2, E3 individuals, typically young, typically in their 50s, sometimes 40s, or into their early to mid 60s when symptoms began. And interestingly, it's typically not amnestic, at least as the major feature. They do have memory issues, but they have other issues such as dyscalculia and agraphia, anomias, partial Gerstmanns, things like that, other things that are associated with cortical presentations for Alzheimer's instead of hippocampal presentations. And when you look at their studies, this supports this notion because the studies show abnormalities outside of the typical region of abnormalities for Alzheimer's, or in addition to the typical regions. And what's been interesting, so far these people are turning out to have a different underlying problem, which in many, many cases seems to be exposure to unrecognized toxic insults in these people. So we're very interested in looking at, as you said, ApoE4 and the genetics, which give you some idea about what type you actually have. But I think looking at these various types has given us new insights into being able to break these down. So, for example, from now, when you're starting to look at drug trials, you can say, okay, we're going to use a drug in this type or that type, this subgroup or that subgroup, rather than across the board where you may be looking at a mixed bag.
That's very interesting. So it's just like in cancer where you have essentially molecular subgroups of different types of cancer that are treated very differently and come from very different mechanisms and have very different symptomology and everything down the line. So you're saying the same thing for Alzheimer's disease, essentially.
Exactly.
Interesting. Very interesting. So you were talking before about looking at clinical research trials, and typically clinical research trials are developed to look at one factor at a time, not multiple therapeutic interventions that are all combined for an optimal approach for one individual. So I can imagine trying to pass the proposed treatment regimen through either a funding agency or an institutional review board can be a little bit tricky. Have you had any pushback with respect to clinical trial approval due to the complexity of your approach and looking at multiple factors instead of changing one factor at a time?
Yes, that's exactly been the problem. So thank you for asking that. As you know, trials are set up to alter one variable, and the idea of multivariable trials is not something that has been well accepted yet. But certainly, with the changes in computation, this should be something that should be readily addressable in years to come. And I think it's something that's going to be important. If we are going to optimize our treatment for these chronic illnesses, then number one, we have to look at multivariable trials. And number two, we have to look at personalized trials. And this is, as you know, this is the problem. The idea of a one-size-fits-all is not going to be optimal for any person. You need to look at personalized approaches. So this all started, the MEND approach all started because back in 2011, we proposed the trial, the first comprehensive trial in history. And the idea was to take a new drug candidate that's called tropicitron that we had identified in our drug development studies in the laboratory as being a very, very good candidate. In fact, we published information showing that it is better in transgenic mouse models than either donepezil or memantine. And so we wanted to use this with and without the entire system, where we would change all these other things that we talked about earlier, all the metabolic parameters. And so the idea was, it was a double-blind, placebo-controlled trial where you'd have placebo only, drug candidate only, and then whole system with placebo and whole system with drug. And the hope was that we would be able to see much better effects with the drug when you had the other parameters affected as well. And the response we got back from both the public and the private IRB in Australia was that this is not the way you do clinical trials. You don't change multivariables at once. So they would not allow us to do this. And we're now in trial with the one drug alone, which they would allow us to do. But clearly, this is not the optimal way to go after this underlying pathophysiology. And I think that's becoming more and more clear. So that is one of the big issues at the moment. How do we go forward with the idea of multivariable trials and get IRBs to understand that that is the way to address chronic illnesses?
It's like you said, especially with the push for personalized or precision medicine that's going to be necessary. So essentially it's going to be reinventing the IRB wheel.
Right. And if you look at what Lee Hood has called P4 medicine, I think this is very much along the same lines. Lee and his group at the Institute for Systems Biology are looking at many, many different variables. And I think that this supports the notion that ultimately, if you're going to want to change aging-related diseases such as Alzheimer's and cardiovascular disease and cancer, you're going to have to consider all these many variables, just as we are doing currently.
Do you think this approach, the MEND approach, will be useful for other types of neurodegenerative disease processes? Or do you think there are other diseases, even not just neurodegenerative diseases, but other diseases we should be looking at with respect to a multimodal approach?
Yes, we've had many people write to ask about that. What about Parkinson's? What about Lewy body dementia? What about frontotemporal? And then what about, as you say, other systemic illnesses? In general, I think that this sort of approach will be the way to go after chronic illnesses. And if you look at the functional medicine movement started by Dr. Jeffrey Bland and others who are interested in that sort of work, such as Dr. Mark Hyman, and of course there are many others, they've come to this same sort of conclusion, that in fact, you look at the biochemical parameters that drive the underlying problem, then you can affect those biochemical parameters in a coordinated and extensive way, and get improvements that you could not otherwise get with single modal, monomodal approaches. And these monotherapeutic approaches are really 20th century approaches when it comes to chronic illness. Now, one of the big caveats is with Alzheimer's disease, it does appear that metabolic inputs are critical. It does appear that whether you have insulin resistance, whether you're carrying a high homocysteine, whether you have trophic withdrawal and hormonal withdrawal, these things all do seem to be important for the underlying process. Will that be the case in these other neurodegenerative illnesses? And it's not clear yet. Clearly, if you look at Parkinson's, obviously there's been a lot written about effects on mitochondrial complex 1, and you can obviously recreate the disease just by impacting that one complex. But for someone who has the disease, what are you going to have to impact? And certainly in some of these others, for example, Huntington's, this is really a genetic problem. So will there be a ways to get after this? I think this is an open question right now.
Yeah, I think it's going to be really interesting to see. But I think it's going to be in the next 10, 20 years, it's going to be interesting to watch how medicine approaches this, multimodal, systemic approach to a lot of these disease processes instead of looking at things as a one target entity. It's going to be looking at things from a body-wide perspective.
Absolutely. And if you go back to the early 1800s, Dr. Ignaz Semmelweis was looking at why it is that women would die following childbirth when they were when they were delivered by one group, but not the other group. And of course, they had very low mortality when they were delivered by the midwives, and they had a very high mortality when they were delivered by the medical students. And everybody understood that, and people would actually actively stay out of that the group where there was a high mortality. And he ultimately realized it was because the medical students had been working on cadavers and were transferring something. And at the time, it really wasn't understood what that was all about, but they were transferring something. And he, of course, developed the hypochlorite approach to preventing this, and he was able to drop the mortality to almost zero. Very striking improvement. And if you read the comments of the experts of that era, where they said things like, it simply can't be that something that's a tiny amount of something under your fingernails could make such a big impact on someone who is having a baby and dying from this puerperal fever. You realize that we all have certain ways that we learn medicine, and we learn specific concepts, and it's often difficult to give up those concepts. We're expecting a certain approach, and I read every day someone saying, where's the silver bullet for Alzheimer's disease? And so it's hard, I think, for us to give up our cherished concepts. We're expecting the silver bullet approach, and maybe, in fact, we need a silver buckshot, not silver bullet.
I like that. Yeah. So instead of looking at one target and then whittling it down, doing cell culture studies and animal studies and everything over the course of about a decade or 2 decades, researching that one target, maybe we should be looking at things from a different perspective. Yeah.
And again, that comes back to what you said earlier, precision medicine. Mm-hmm. You have to look at what's driving the process and then address all of the things that are driving the underlying process.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
So you were talking about, we've talked about genetic risk factors and other things that are risk factors. So exposure to toxins earlier in life or even later in life, head injuries. So say you have an individual and you know that they're at increased risk of developing Alzheimer's disease. Do you foresee your approach as being something that they can take into account and implement in their lifestyle to try to minimize their risk of developing Alzheimer's disease?
Absolutely. And of course, I think many people are coming to the conclusion that we should be looking earlier in life and we should be making an impact. I mean, I think it's highly likely that if we initiated this globally, you could dramatically reduce the global burden of dementia. And for example, there are 75 million Americans who are ApoE4 positive. The vast majority, as you know, do not know that they're ApoE4 positive. And so let's imagine that there was a national program saying, look, go out, get your genome tested. That's an easy thing to do right now, and get on the program so that you reduce your risk going forward. And then you can check every year or two and see where you stand metabolically and see what your likelihood is of developing this. And if people would embrace that sort of approach, we could dramatically decrease the likelihood. Right now, your overall groups, your likelihood of getting Alzheimer's during your lifetime in the United States is about 15%. This could be reduced dramatically if people would look earlier on and not wait until symptoms appear, or when symptoms appear, come in early. Don't wait. Everything right now is directed toward the exact way that's the opposite of what you would like. People are pushed to wait longer and longer. They want to stay at work as long as possible, instead of coming in and getting the problem fixed so that they can, do better with their job. We've had a number of people who've been struggling at work who on the program have done very well and are back at work full time, including that first woman that I mentioned to you earlier. I think that's the way of the future.
Yeah, I think, it's really interesting you say that, because I think up to this point, you find out that you're E4, so what? So then what do you do? But now you guys actually have an approach, something that people can do and implement, and, factors that they can look at and, like you said, measure and keep track of over a course of time and make sure that they're doing everything they can to minimize the risk of developing Alzheimer's disease. It's just like, early detection for breast cancer or prostate cancer. Before, if you didn't have any kind of approach to actually handling it, it'd be, it's like, okay, well, then you're just going to run around scared or concerned that you're going to develop it. But if you actually have something that you can do about it, then that's a game changer.
Absolutely. And the thing is, it's not just changing those variables, but it's also following them. So you can say, aha, I've done really well with these three variables, but I see, for example, this fourth one, I've not impacted enough yet. Okay, so what do I need to add? That's one of the parts about this 21st century medicine. It's not get a pill and go home. It's make some changes, come back and see where you stand, and then iterate and continue to tweak and optimize over time.
Yeah, not just personalized or precision, but evolving too with the patient.
Exactly.
Yeah. So are there any plans to do longitudinal studies? You talked about looking at younger people and kind of following them down the line. Do you have any plans to do studies like that?
So right now we're looking at people who are in early symptomatic stages, but certainly there are many groups interested in prevention and looking at that longitudinally, and that will certainly be the way of the future. The first thing we have to do, though, is to get an IRB that is open to the notion of multivariable clinical trials.
Mm-hmm. Makes sense. So that's the next step then. You said you're going to be looking at more patients and just trying to up the N, essentially, and trying to optimize and optimize the treatment regimen as well.
And one of the big issues is what happens to the pathophysiology. So when people get better symptomatically, what do you actually see? Does the amyloid go away? These are the sorts of things we're exploring right now. We, as I mentioned earlier, have found that you can see dramatic changes in volumetrics on MRIs. So we want to understand what is actually changing in the process. Do you heal things, or are you simply functioning better with the same problems? How long can it be sustained? What do you have to do to sustain it? All these sorts of things.
Yeah. Well, it's going to be really interesting to follow your work and see where things go down the line. I'm going to ask you a personal question. What got you interested in this area of research in the first place? Is there something specific that made you decide to look at Alzheimer's disease?
Yeah, when I was a freshman at Caltech, I read a book that's called Machinery of the Brain by Dean Wooldridge. And I was hooked. I got so excited about how the brain works and the various changes that can occur, from status epilepticus to loss of memory. Then when I was an undergrad there, Seymour Benzer was doing this just amazing work where he was identifying things like the dunce fruit fly, where you could actually have a Drosophila that could not learn new information based on a single gene mutation. And I just thought that was absolutely fascinating. And so I wanted to understand.
It's called the dunce?
Dunce.
That's
yeah.
funny. Okay.
DUNCE. Well-described fruit fly, and it turns out to be in phosphodiesterase. So it is a cyclic AMP-related pathway, and much like what later Eric Kandel and his group worked out as being critical for memory formation and for plasticity. Then when I started learning about the brain and how it worked, I really wanted to go to medical school to understand what actually goes wrong when you lose your ability to do learning and memory. What actually happens? And when you get hit in the head, what happens? And when you get Huntington's disease, what happens? And all these sorts of things. And so, wanted to understand what is the underlying pathophysiology. How do these things actually work? What's the big picture of what's driving these? And I think what's been most fascinating to me is what causes the diseases, and the molecular neuroscience revolution has actually allowed us, just with things like ApoE4, it has allowed us to open the hood, look under the hood to see what is actually driving these problems. But the thing that's come out of this is that there is a schism in medical training and in the standard of care for medicine between 20th century medicine, where you ask what the problem is, and if it's hypertension, you give an antihypertensive, and 21st century medicine, where you, as you said, precision medicine, where you ask, why is it? So why did you get hypertension? And let's look upstream instead of just giving you an antihypertensive and sending you home.
Mm-hmm. Yeah. So, and like you said, with the advent of the technology that we have today, you were saying it gives us a chance to look under the hood and see what's going on. It also tells us what we should be looking at, or at least gives us a better picture of what we should be looking at specifically.
Absolutely, and it begins to tell you. But when you look at the pathophysiology, it doesn't necessarily direct you toward a single pill. And I think that the approach of taking the overall metabolic approach will be the foundation on which future drug discovery and drug testing will be based. So absolutely, drugs are critical. They're important. They're effective, but you have to have the right background on which to use them.
Mm-hmm. So I have to say, your research is relatively paradigm shifting with respect to looking at Alzheimer's disease, as you refer to it as programmatic downsizing and this multimodal approach to treating Alzheimer's disease or even preventing it. So were there any aha moments that you had along the way that were just kind of groundbreaking, the light lit up and you said, wow, this is really, really interesting, or this tells me what I was expecting to see, or this is telling me something I wasn't expecting to see?
Yeah, I mean, we've had a couple of them. The first aha moment was when we were looking at a receptor that was supposed to be involved in supporting neural growth. And we found out that it induced the death of the neurons, the opposite of what it was supposed to be doing. And that was the first aha moment. We realized, this is backwards. This is essentially killing the cell unless it gets the support that it needs. And that was the birth of dependence receptors. And then when we saw that A beta is really it really functions as an antitrophin. And we realized, okay, so when you have 1,000 times too much of this in your brain, which you do in Alzheimer's disease, no big surprise that you get an antitrophic syndrome. And then, of course, another big aha moment was when the woman called me at my home on Saturday after 3 months on Mend. And at the time, it wasn't even called Mend. We didn't know anything about this. And she said, my gosh, I'm back at work full time. I'm completely back to normal. In fact, I'm better than I've been for decades. And so I thought, okay, it's not just transgenic mice. It really does work. This really is the way things work in Alzheimer's disease. And then the other aha moment was when we saw the first MRI that showed that, oh my gosh, the brain is actually, it's increased in volume. The guy who actually had that done was arguing with me, why are you even doing this? Because everybody knows your brain's not going to get bigger when you have Alzheimer's disease. And this guy has well-documented ApoE4 positive, amyloid positive, PET scan positive, everything, and dramatic changes. So these are the sorts of things that, kind of change course and tell you, things there. Whatever is going to be the final answer here has to be going down this pathway in one way or another.
Yeah. It has to be just absolutely fascinating. Like you said, this is it's just such a different approach. So probably every time you turn a corner, it's something new, and it's insight into the disease process that hasn't been discovered yet. So that's absolutely fascinating.
Thank you.
So, quick question for you: the institute that you work at, can you describe the work environment there?
Sure. So I actually split time between two places. I'm at UCLA at the Easton Laboratories for neurodegeneration research, and then I am also at the Buck Institute for Research on Aging, where I was founding president and CEO back in 1998, and I have part of my laboratory back at the Buck Institute for Research on Aging, which is the nation's only independent research institute focused on aging and age-related diseases.
Is it predominantly MDs, PhDs, students?
Right. So in both laboratories, it's predominantly PhDs. And yes, we have students come through all the time. We have had summer students, et cetera. We've had some graduate students, and there are others as well. But it's predominantly PhDs.
Okay. And do you run animal studies there as well?
Yes, we typically do transgenic mouse studies. We do studies on cell culture models. We've been working on cell culture models of neurodegeneration from the very beginning. And in fact, one of the aha moments we published back in 1995, showing that neurodegeneration-associated genes, when transfected into neural cells, increase the risk for them to undergo apoptosis. So, and this has turned out to be true for all of the neurodegeneration-associated genes. It somehow increases the likelihood that these cells will commit suicide. And we were really excited and surprised to find that, because at the time it was said that anything that had a short-term effect in cell culture couldn't be related to a chronic illness in a human being. Fortunately, that turned out to be wrong.
So one more personal question. So you know all these different factors that potentially affect your risk of developing Alzheimer's disease. Do you implement all of those into your lifestyle personally as well?
Yeah, great question. So I try to. And of course, I struggle with some of them. Definitely, as Americans, we typically have a relatively high simple carbohydrate diet, just from the various processed foods. So, in fact, I've changed my diet over the years with help from my wife, who is an integrative physician. So she's better than I am and teaching me a lot of this stuff. And what's been one of the fascinating things is that the basic neuroscience coming out of the test tube has interacted perfectly with the integrative medicine and has told us about some of the underlying mechanisms. I used to laugh at integrative medicine 20 years ago, and I'm not laughing anymore. I should have listened to my wife back then.
You don't tell her that, though.
So I'm trying to do many of these, and definitely I have more work. Definitely trying to do some of the lifestyle issues. One of the big issues that comes up again and again: stress and sleep. And these things unquestionably impact the Alzheimer's related balance. And one of the common things that we see in these people who come through is that on top of all the other metabolic parameters that they have shifted, they are too low on their sleep and too high on their stress. And as they're getting better, one of the things that helps them is to address both of those. And I'm not very good about addressing either one of those yet, and especially the sleep one, because, hey, when do you do your emails if it's not at 3 a.m., right? So yes, I'm trying to implement these things, but I've got a long way to go.
Well, this has been really interesting, and I'm very excited about, seeing your recent publication that you said it just came out, correct? About a week ago?
Yeah, so in Aging.
Okay, in Aging.
The same one that we published, Mend, Aging. We published Mend and Aging in September. Aging is it's got a high impact for the field of aging, the highest for the field of aging, and also is is good about getting things out relatively quickly. So I've been impressed with their professionalism.
Okay, great. Well, I look forward to reading that. So, well, Dale, thank you so very much for joining us today on the podcast.
Thank you. I look forward to talking to you again. Take care.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Lots of great information that Dr. Bredesen shared with us on STEM Talk.
I'm very impressed with his perspective of Alzheimer's as fundamentally being a metabolic disorder. His work shows great promise against an otherwise relatively bleak landscape of ineffective Alzheimer's treatments. Most of the research on Alzheimer's is focused on trying to identify a single mechanism rather than on the synaptic network itself. Rather than looking for a single silver bullet, he is taking a multifaceted systemic approach aimed at addressing root causes, of which there seem to be many.
And he is thinking about Alzheimer's more like we now understand other chronic disorders, such as diabetes. His group has had striking results with many patients demonstrating significant improvement. They've even seen substantial MRI volumetric improvement in regions of the brain. Dale's work is fascinating, and I could have talked with him for hours, but unfortunately, all good things must come to an end. I invite you to visit the STEM Talk webpage where one can find the show notes for each episode. You can find us at stemtalk.us. This is Dawn Carnegie signing off for now.
And this is Ken Ford saying goodbye until next time.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.